Today we're examining a central challenge in drug development: how to select the right biological target. The initial choice of a target is a critical determinant of a drug program's success. We'll be looking at how advanced computational and genomic approaches are being used to improve this selection process, based on a recent review in Nature Reviews Genetics.
To begin, let's consider the scale of the problem. Approximately 90% of drug candidates that enter clinical trials fail to gain market approval. This high attrition rate represents a substantial investment of resources that does not culminate in new therapies. It is the primary bottleneck in modern drug development.
So, what is the reason for this high failure rate? The data indicates that the most significant factor is the initial selection of the biological target. Roughly half of all clinical trial failures are attributed to a lack of efficacy. This suggests that the issue often lies not with the therapeutic molecule, but with the validity of the target it was designed to modulate.
To understand how these challenges are being addressed, we'll structure this analysis into 5 sections. First, we'll define the target selection problem in more detail. Second, we'll review the use of human genetics as a source of evidence. Third, we'll discuss how computational methods augment this data. Fourth, we'll examine frameworks for integrating multiple lines of evidence. And finally, we'll outline a proposed unified probabilistic framework for target selection.
So, how can we improve the process of target selection? A leading strategy is to anchor the process in human genetics. The central hypothesis is that by using genetic data to guide target choice, we can de-risk drug development from its earliest stages, well before significant investment in chemistry and clinical studies begins.
The concept of experiments of nature is key here. Genetic variation within the human population results in differing levels of protein function or expression. By studying the association between these naturally occurring variations and disease outcomes, we can, in effect, observe the lifelong consequences of modulating a specific protein. This provides a human-based in vivo model for the potential effects of a therapeutic intervention.
This approach is supported by empirical evidence. Retrospective analyses of drug development pipelines have shown a clear trend. Drug targets with supporting human genetic evidence are 2.6 times more likely to progress from phase 1 trials to market approval compared to those without such evidence. This statistic provides a strong quantitative argument for integrating genetics into the foundation of drug discovery.
Accepting that genetics can guide target selection, the next challenge is methodological. How do we progress from a statistical association signal in the genome to the specific effector gene and causal mechanism? Let's examine the techniques used for this purpose.
The process typically starts with a genome-wide association study, or GWS, which identifies loci associated with a disease. From there, 2 main strategies are employed. A variant-centric approach focuses on the likely causal variant itself, using its genomic context and functional annotations to link it to a specific gene. A gene-centric approach, conversely, evaluates all genes within the associated locus, prioritizing them based on their biological plausibility and other converging lines of evidence.
To infer causality from these associations, a technique known as Mendelian randomization, or MR, is frequently used. MR leverages genetic variants that reliably predict an exposure, like the expression level of a protein, as an instrumental variable. This allows one to estimate the causal effect of that exposure on a disease outcome in a way that is less susceptible to confounding than traditional observational studies. One must, however, account for potential violations of MR assumptions, such as horizontal pleiotropy, where a variant influences multiple unrelated pathways.
A practical application of these principles can be seen in the development of TYK2 inhibitors. A GWAS identified a variant in the TYK2 gene that conferred protection against several autoimmune diseases. Functional studies revealed that this variant reduces the kinase's activity. This genetic finding provided a clear therapeutic hypothesis. Pharmacological inhibition of TYK2 could mimic this protective effect. This directly informed the development of deucravacitinib. Further confidence was built using methods like Mendelian randomization and transcriptomic analyses, which reinforced TYK as the correct target.
Another informative concept is the allelic series. This refers to the existence of multiple variants within the same gene that have different effect sizes and population frequencies. By studying individuals across the spectrum, from common, low-effect variants to very rare high-impact mutations, we can construct a naturally occurring dose-response curve. This helps to define the therapeutic window, indicating how much target modulation is needed for efficacy and at what point adverse effects might emerge.
While genetic signals provide a strong foundation, their detection can be limited by statistical power.
This is where computational methods, particularly machine learning, become valuable. These techniques can enhance the underlying data to increase the power of genetic discovery. Machine learning contributes to more precise phenotyping in several ways. It can be used for phenotype extraction, for example, by identifying disease-relevant features from medical imaging data. It's also used for phenotype imputation, filling in missing data points to increase sample size. Furthermore, it allows for phenotype augmentation, which can transform binary case control labels into a continuous representation of disease severity, often better reflecting the underlying biology.
For rare genetic variants, individual analysis often lacks sufficient statistical power. The solution is aggregate variant testing. These statistical methods group together multiple rare variants within a defined genomic unit, typically a gene. By analyzing their combined effect, these tests can detect associations that would be missed at the single variant level. The choice of test depends on the assumed genetic architecture of the trait.
We now have multiple streams of evidence, from GWAS and rare variant studies to functional genomics. The next operational question is how to synthesize these disparate data types into a coherent prioritization of potential drug targets. This requires a formal framework for evidence integration and weighting.
Several computational frameworks exist for this purpose. Some, like the Open Targets platform, aggregate data from a wide array of sources and use a weighted sum approach to generate a score. Others, such as the genetic priority score, use machine learning models trained on known drug successes to empirically learn the optimal weights for different evidence types. Each of these approaches has distinct assumptions and strengths. A fundamental distinction exists between tabular and network-based models. Tabular approaches evaluate each gene-disease relationship in isolation, based on the direct evidence available for that pair. Network-based approaches, in contrast, model the broader biological context, such as protein-protein interaction networks. This allows them to identify targets that may lack direct genetic evidence themselves, but are located in close proximity to other disease-relevant genes within the network. This leads to the central proposal of the review, the development of a single, unified probabilistic framework. The goal is to integrate these different methodologies into one systematic end-to-end process for target identification and prioritization.
This proposed framework consists of 4 sequential stages. First, machine learning is used to generate deep and accurate disease phenotypes from large-scale biobanks. Second, a comprehensive suite of genetic methods is applied to compile all available gene disease evidence. Third, this evidence is integrated within a probabilistic model that assigns a confidence score to each potential target. Finally, these scores are combined with data on target tractability to generate a ranked list of candidates for experimental follow-up.
Realizing this vision requires addressing several significant challenges. There's a persistent need for larger and more ancestrally diverse genomic datasets. Data sharing and harmonization across public and private entities must be improved. These methods also need to be applied more broadly to areas of unmet medical need. Finally, a key future direction is the development of models that can predict not only efficacy, but also potential adverse effects associated with a target.
This leads to the final question. Can a unified, probabilistic framework substantially mitigate the biological and financial risks inherent in drug discovery? The evidence suggests that we are in the midst of a fundamental shift, moving away from a process characterized by trial and error and towards one that is systematic, quantitative, and grounded in human genetics. While challenges remain, this data-driven approach offers a clear path toward improving the efficiency and success rate of developing new medicines.